Today we begin with the chapter nutrition. It is the process of taking in food and utilizing. So nutrition is a process of taking food and utilizing it for energy requirements. A nutrient is a substance which an organism obtains from its surroundings and uses it as a source of energy. You know that food is needed by all living organisms to perform different activities like growth, repair, for maintaining body temperature, and to perform various types of other activities. And the mode of nutrition varies in different cells and in different organisms. The classification of nutrition is mainly of two types, into two groups. It is autotrophic and heterotrophic. The examples of autotrophic nutrition include green plants and blue green algae. The heterotrophic nutrition is again subclassified into saprophytic, parasitic, and holozoic. The examples of saprophytic include fungi and yeast. Parasitic include cuscuta and plasmodium, and the holozoic examples include man, dog, amoeba, and some other animals. We will study in detail about these types. Coming to the autotrophic nutrition, the name itself implies that auto means self, means these organisms can prepare their own food material by using inorganic substances like carbon dioxide and water to produce complex compounds such as carbohydrates, example glucose, proteins, and lipids. So, the compounds which are produced here during the respiration process are used during the respiration process to yield energy rich compounds such as ATP, which will be utilized to perform different types of activities. So, the green plants contain a green pigment called chlorophyll, which is located in an organelle called chloroplast, which is capable of trapping sunlight energy. The process in which the green plants make their own food material utilizing inorganic compounds such as CO2 and water in the presence of sunlight using the chlorophyll pigment, it is termed as photosynthesis. So photosynthesis is a process by which plants prepare their own food material. This is the chemical equation of the process photosynthesis. Here, the six molecules of carbon dioxide are combined with six molecules of water to yield one molecule of glucose, and along with it, six molecules of oxygen are also released into the atmosphere. Here are some activities. First, to detect the presence of starch in leaves, we have to take a leaf and boil it in methylated spirit over a water bath till it becomes pale white due to the removal of chlorophyll. Then we add a few drops of tincture iodine or betadine solution. If starch is present in that leaf, it will be indicated by a blue-black color. If starch is not there, it will not be indicated. So when we do the same test by covering the leaf with a black paper with certain type of shape or an alphabetic shape with some type of design, when we cut and we fix it properly such that the light does not enter through the part of the paper, and when we test and when we leave the leaf for some time, and again after that when we test for the presence of starch, we will find out that the part which is exposed to sunlight will indicate the presence of starch, will show positive for presence of starch, and the part which is covered by the paper shows negative, indicating the absence of starch. Coming to Mole's half-life experiment, we need a destarched plant, so we will keep a potted plant in dark for 2 to 3 days, and here a part of leaf is kept enclosed. Here the part of leaf is kept enclosed in potassium hydroxide solution, because potassium hydroxide has the capability to absorb carbon dioxide. So here in this part of the leaf cannot get carbon dioxide because it is enclosed in a jar containing potassium hydroxide. So when we test after some time for the presence of starch in this leaf, the part of the leaf which is exposed to sunlight, means which is not present in the test tube, will show positive for the starch, and the part of the leaf which is present in the test tube will show negative, indicating the absence of starch. Coming to the early experiments, Joseph Priestley performed a series of experiments that revealed the essential role of air in the growth of green plants. You may recall that Priestley has discovered oxygen in 1774. Priestley